Let's go ahead and get started for the day. Let me begin with just a brief review of a few of the key ideas from the last lecture, and then I'm going to cover the material that didn't get finished on Wednesday, and we'll have a quiz, and then we'll move into the topics for today's lecture. But remember that we talked about the components of Darwin's theory of evolution by natural selection, and they're just key ideas that motivated this development of this really revolutionary theory about the origins of biodiversity. And one of the key ideas is that the physical world changes slowly but steadily, and that organisms have progressively adapted to those changes over time. And that was embodied in the geological theory of Charles Lyell of the 19th century, which emphasized a concept called uniformitarianism. Uniformitarianism simply claimed that the geological world has changed slowly owing to the action of forces that occur every day, like wind, erosion, storms, and so forth, which gradually lead to the deposition of silt and other materials in bays and estuaries, or other kinds of geologic forces that are more sudden, like earthquakes and volcanoes, but those also are observed in the span of a human lifetime. So the kinds of forces that we would observe in the span of the human lifetime, when accumulated over long periods of time, lead to geologic change and changes in the physical world. The other key idea was common descent, that all of the organisms alive on the earth today are related through common descent. And we now know that that idea is very, very firmly established, because virtually all life on Earth uses the same system of heredity. It uses DNA, or in the case of some viruses, RNA, as the basic system of heredity, establishing beyond any reasonable question the notion of common descent, a common history back to a single origin of all life on Earth. The process responsible for the elaboration of biological diversity is natural selection, and we're going to talk more about natural selection during today's lecture. Finally, over long periods of time, natural selection can lead to the origin of new species, and we'll have a whole lecture on how that happens in another week or so. So what are the processes of natural selection? They are pretty straightforward. The first fundamental postulate is that populations vary genetically. That is, the individuals within a population differ genetically. And we know this is the case. We have all kinds of powerful tools now that allow us to look at the genetics of different individuals of almost any species, and it's clear that there are mutational differences among individuals. So individuals in a population vary for many traits. Some of that variation is heritable. It's caused by genes. The traits that happen to confer better adaptation in a particular environment will be those that are naturally selected, and the genes that determine those traits will be disproportionately transmitted to future generations. So that's the key concepts. It's very simple. And then we went on to talk about the fact that evidence for evolution can be found in many different lines of evidence that support the notion of evolution, including a very extensive fossil record, which is still being elaborated. Even in the last year or so, a new fossil species of Homo was discovered, which was at approximately 4 million years old, fills in one of the key elements in the evolutionary lineage that led to man. So there's quite a lot of extant work on discovering and documenting fossils. Extinction, I mentioned in the last lecture that 99% of all of the species that have ever existed on the Earth have gone extinct. 99% of all species have gone extinct. That extinction is a normal process of the sort of biological history of the Earth, and that the complement to that process is speciation, the origin of new species. So there's this constant flux of extinction versus speciation. The average lifetime of a species on Earth, as deduced from the fossil record, is about 2 million years. We looked at things like homologies. We identified several kinds: genetic, developmental, and structural homologies, all providing different kinds of evidence of descent from a common ancestor. And then we looked at evidence for ongoing genetic change in the context of evolution to antibiotics, and we used a specific example, and that was the evolution of the bacterium which causes tuberculosis in response to the antibiotic rimfampin, and we talked about a specific case of an individual who died because of the evolution of antibiotic resistance. So what are the key points then? First, evolution by natural selection is a very simple idea. It occurs just like objects falling from gravity. It occurs naturally whenever there is heritable variation that leads to differential reproductive success, period. And that's inevitable when there's a large reproductive excess, when more potential individuals can be born than will survive. Natural selection is acts on individuals. Individuals survive or reproduce. But evolutionary change is a property of populations. It's a collection. It's an emergent property that arises at the population level. Individuals don't evolve. They either survive and reproduce, or they do not survive and reproduce. It is the population whose composition changes over time. It's a key concept, and it certainly will appear at some point in a question on a quiz or exam. Evolution is defined at its simplest and most elemental level as simply a change in allele frequencies, and we'll come back to that and amplify on it today and on Monday. Fitness is the ability of an individual to produce offspring relative to other individuals in the same population. And adaptation is a heritable trait that increases an individual's fitness in a specific environment. It's environment dependent. Adaptations are dependent upon the particular environment you live in, just as for the tuberculum bacterium, having the rimfampin serine to leucine amino acid substitution in the small subunit of the ribosomal RNA molecule was an adaptation to the environment when the antibiotic was present. Okay, so those are some of the basic ideas. Now I want to pick up with what we didn't cover in the last lecture very quickly. A question you may ask is, well, okay, so antibiotic resistance or insecticide resistance are special cases. These involve human changes in the environment. What about in the natural world? Can we document evolutionary change in short periods of time by direct observation in the natural environment? And the answer is yes, we can, and there have been many examples documenting evolutionary change in the natural environment. Your book talks about one example, which is work of a husband and wife team named Rosemary and Peter Grant, who were both on the faculty at Princeton University, who've worked for 40 years studying the evolution of ground finches in the Galapagos Islands. And they've focused much of their work on a particular island, a small island. It's just several acres in extent. And they've studied the medium ground finch, which is shown here in this panel, and a picture of the island is shown in the bottom panel. And over the years, they've shown that many different phenotypic traits have a heritable component in these ground finches. Among them, body size and the beak length and structure. And so we're going to focus on the beak length and structure. And early in their research, a remarkable, or not necessarily remarkable, but an interesting environmental perturbation occurred. There was an extreme drought. And the extreme drought lasted for a considerable period of time. And during that period, this was on this one island, the Galapagos. And during that period of time, the ground finch population collapsed. 84% of the population died. And they were able to monitor the birds by tagging them. They took careful phenotypic measures on all of the birds. So in one season, the population collapsed, 84% of them died. They were able to recover some carcasses of dead individuals and infer that they died from starvation because the drought killed the seed-producing plants that the ground finches depended on for their sustenance. So the question they asked is, were there changes in the beak size and shape caused by this crisis in food supply? And why did they ask that? Well, because the beak is exquisitely adapted to opening the seed that the finch feed off. But when the seed-bearing plants died or failed to produce enough seed, some of the finches were able to shift to another resource, a berry on a perennial shrub on the island, and use that as a temporary food source. And if the grants measured the beak length and width, and then they already had measures prior to the drought, shown in this first histogram, and then they also measured it among the survivors after the drought and showed that there was a statistically significant shift in the size of the beak towards greater depth, which turned out to be an adaptation for breaking open these tough berries so that the few individuals in the population, which by natural variation had larger and stronger beaks, had an advantage in shifting to this new food source. They survived, left their genes behind, and there was a shift in beak length. Now, grants continued to work and observe these populations, and several years later, there was a year which had very high levels of rainfall. The seed crop was abundant. And under these circumstances, natural selection actually shifted beak size back towards the smaller beak because it had a greater leverage, a greater lever power in opening seeds. So that illustrates that you can observe natural selection on phenotypic traits in well-designed experiments in nature where humans are not an intervening agency. It also demonstrates another very key point, and that is that natural selection is not goal-directed. There's not an end point. It's simply the opportunistic change according to what's adapted in the particular environment at a particular point in time. So at one point in time, under extreme drought, larger beaks, which facilitated a shift in food resources, were selected for and were adaptive. But when the wetter conditions returned, Smaller beaks were adaptive, and selection reversed itself and went back in the other direction. So this leads into a few points about misconceptions and inefficiencies in natural selection. The first is that, as I've just said, evolution is not goal-directed. It's whatever adapts individuals to a particular environment at a particular point in time. It is not progressive. There's not as envisaged by early 19th century scholars, there was a notion of a sort of a ladder, a progressive ladder of life that led naturally upwards to higher and higher levels of complexity. Well, that's not correct. It's a wrong notion. There is not a progressive aspect to evolutionary change. And we see lots of examples of the loss of important organs, systems in environmental change. An example is blind cave fish. These are fish which have colonized caves, no longer have a need for sight. Eventually, the genes that determine eye structure and function mutate into non-functional forms, and you end up with blind populations because the eye adaptation is not of use in that environment. Another key point that we'll come back to in a couple of slides is that organisms do not act in a way that is good for the whole species. Selection acts to simply maximize reproductive success, and whatever genes do that will be selected. And then finally, there are changes in individual phenotypes in response to environments which are not heritable. And a great example is acclimations. There are lots of different kinds of acclimations. We tan when we sit in the sun. It's an acclimation. It's a reversible change in phenotype that has nothing to do with heritability. Another very good example of an acclimation is response to high altitudes. When we spend a few days at a high altitude, those of you who like to ski know this well, the first day or so it's hard to breathe and you're tired. After several days you develop more hemagglutinin, better oxygen binding capabilities. The high altitude effect is diminished. And this is just some pictures from La Paz, Bolivia. It's one of the highest national capitals in the world at almost 10,000 feet. People acclimate to walking in these high environments. Now, this question about selfish genes. This is a Gary Larson cartoon, which I think makes this point particularly well. There was a kind of a piece of folklore about lemming behavior, that lemmings, when their populations get too large, rush into the sea and drown in order to diminish the population for the good of the species. Well, it's hogwash. Lemmings don't do that. They do migrate when population density gets large, but they don't commit suicide. And in fact, the point of the cartoon is that if you have one individual that's cheating, the individual with the inner tube who's not going to drown, that's the individual that's going to leave its genes behind. And so under most circumstances, natural selection will favor selfish behavior that leads to the transmission of one's genes. Now that seems a little bit morally repugnant from a human point of view, but that's the way the mechanism works. Maybe it's not repugnant if you work on Wall Street, but a lot of people are worried about, well, we know that there are behaviors that appear to benefit the group at the expense of the individual. You see this sometimes in wartime, but there are examples of, say, prairie dogs that give a warning call when a threat is present, therefore calling attention to the individual that gives the warning call while other individuals can hide. And there's an explanation for that kind of behavior, and it's in an area called kin selection. If you're part of a related family group and your behavior gives a large enough advantage to your relatives that the net transmission of genes will be increased, then that behavior can be favored. But generally, selfish genes are favored. So what are the limitations of natural selection in a genetic context? Well, there are several. One comes up in the context of what are called genetic correlations. Our genes specify our development through time. And individual genes may affect many aspects of phenotype, particularly ones that are very fundamental to development. So if we select for genes that increase body size, they're likely also to increase things like arm length and leg length. Those are correlated responses. And so selecting on a particular trait is likely to cause correlated responses in other related traits that share an underlying genetic program. Of course, if there's no genetic variation at all, you can't have successful natural selection. You can select away on a population, but nothing's going to happen to the next generation because there's no variation to select on. There are what are called historical constraints. These have to do with structures. And think back to the slide I showed you on the evolution of tetrapods, where we began with the lungfish, and eventually, after about 23 million years, we actually ended up with tetrapods that were able to walk on the terrestrial Earth. That pathway through which that adaptation of limbs arose was through the modification of the bones for the dorsal gill. And the historical constraint was that was the available structural pathway that could be exploited as evolution gradually moved towards occupation of the terrestrial environment. So there are historical constraints. There are fitness trade-offs. This is a compromise between traits where one trait may be adapted in one environment, a different trait in another, and you can't optimize both simultaneously. So selection, because it operates on the whole phenotype, we either survive and reproduce or we don't, and each of us is the manifestation of all of the genes that we possess, acts on many traits and many genes at once, and genetic correlations and fitness trade-offs have a big effect. So these are limitations on natural selection. Now, we're going to have our quiz, and hopefully we can get this done easily. I need my, yeah, okay, I'll let you operate that. So what we're going to do is we'll give you 1 minute for each question, which should be more than enough. And
do you want me to demonstrate how to reset it again?
I'm sorry?
Do you want me to demonstrate how to reset it?
Oh, yeah, go ahead. Yeah.
Okay.
Yeah, sure.
I can shout it. I can shout
Yeah,
it.
go ahead. No, that's right.
To reset your eye clicker.
Hey guys, pay attention for 1 sec.
Thanks. To reset your eye clicker, press and hold on the on-off button until it blinks, and then press AA, and you can see a green light. It's not good at all.
Did you guys do that for that? Good.
So are we ready to go?
Raise your hands if you're not ready. Raise your hand. Wait, mine's up. All right.
Is everybody,
Do you have a question?
is that a question or a?
Justin's gonna help you. Do you have a question too? Ada, can you? Can you use the notes for the quiz?
Huh?
Notes.
What?
Open notes and close.
No, it's closed book.
Closed book.
Yeah, close your material in your computers and your iPhones and everything else. This is closed book. Sorry. These are easy questions. By the time you wouldn't be able, we'll give you 45 seconds per question. You wouldn't be able to look up the answer fast enough, so use your head instead. Are we ready to go?
Stationed around now, okay?
Ready to go? Okay, here's the first question. 45 seconds. Okay, second question. Oh, I guess I can't advance this. You've probably got me blocked with that. Okay, second question. Okay. Third question.
Sorry.
Okay, go ahead. It's not, timing isn't going. Okay, so if you want, yeah, so this is the question one. Okay, we'll look at the responses. So let me, well, okay, so you can see how you did. Let's go to question two. Okay, and then question three. Oh, well, okay. So you all did pretty well. Now let me, can we give me back control? Let me just go over the right answers. They're given here, okay? Question one, what was it asks, why are some traits considered vestigial? Identify the correct response. Correct response is number 3, as most of you got. And that's just the definition that came straight out of your book. I like best the one that confers intellectual ability. Number 2, which statement is false? All right, statement 3 is false. That was a little bit tougher, but it's not just survive, it's reproduce, okay? Everybody in the population can survive, but if only some reproduce, that's what matters. It's leaving your genes behind. And I think there was more variation in the response to this one, but this is the list of responses that you should have given. Number 3, the fundamental characteristics of life are, well, motility is not one of them. 2, spontaneous generation sometimes occurs in a nutrient-rich broth. No. Adaptation is a trait that increases the fitness of an individual in a particular environment. True. The tree of life is completely worked out. No, I emphasize that there's lots more research in that area, and there are only 3 domains of life. Okay? So those are the correct responses. Now let me turn now to the subject of today's lecture, which is drawn from chapter 25, and begin by saying that we've emphasized natural selection up until now as the driving force of evolutionary change, but it's not the only force. If we take the definition of evolution that I just gave you as a change in gene frequencies, there are actually several different forces that will change gene frequencies. And so we're going to spend some time in this lecture and on Monday going through them all. But the first, of course, and the most important, and the one that's responsible for the evolution of adaptation is natural selection. But there is also genetic drift, which is just the random change in gene frequencies due to the random sampling of genes in a population for transmission to the next generation. And we'll elaborate on these ideas. Gene flow represents the transmission of genes between populations due to the movement of individuals or the movement of propagules like pollen of seeds, plants. And mutation modifies allele frequencies because it introduces a new allele into the population. So these are the fundamental forces of evolutionary change. There are 4 of them. You should remember them because they're the kind of thing that it's easy to ask test questions about. Now let me begin with a very important early result after the rediscovery of Mendelian inheritance. I think you probably all know that Gregor Mendel wrote his very important papers on particulate inheritance in about 1864. Gregor Mendel, as you probably know, was a monk in an Austrian monastery, fascinated with the question of inheritance, studied the transmission of a number of traits in peas, which he could easily crossbreed, showed that inheritance was particulate. The inherited particles that determined traits like flower color in the case of peas, or whether seeds were wrinkled or not, came to be called genes. We now know, of course, that genes encode, in many cases, proteins, and that that are responsible for metabolism and structure. So we begin then with the particulate nature of inheritance. Now, the amazing thing is that Mendel's work was not appreciated by his own generation. In fact, the biggest problem that Darwin had with his theory of evolution by natural selection is that he didn't have a mechanism for inheritance. And he appreciated that as a problem. And he tended to gravitate towards a blending theory of inheritance, where it was some miscible, like liquid, transmitted from generation to generation, so that the 2 parents would transmit some sort of miscible substance which would combine and blend to produce the offspring. Well, it develops that a blending system of inheritance is completely antithetical to evolution by natural selection, because a blending system of inheritance, and one can show this with a simple mathematical argument, will have the amount of variation in each generation, so that within just a few generations, 2 or 3 or 4, there's no variation left to select on, and you couldn't have evolution by natural selection. So a particulate system was absolutely essential, and Darwin didn't appreciate Mendel's work, nor did any of the other 19th century biologists that read it at that time. And then suddenly, in 1900, at the turn of the century, 3 different investigators, all interested in the subject of inheritance, rediscovered Mendel's work and appreciated its significance and published on it. And very quickly, in 1902, a medical student actually named Sutton postulated that genes must be on chromosomes, because in the 1890s, by the time microscopy had developed enough, it was possible to see that chromosomes were transmitted through gametes to form a fertilized zygote. And so there was the beginnings of what was called the chromosomal theory of inheritance. But 2 guys, again, simultaneously worked out a simple mathematical proof whose real significance is that it shows that under random mating, genetic variance is conserved in a particulate system of inheritance. And this is called the Hardy-Weinberg theorem or model, and it's really simple, and in your book gives some face to it. So to do this, they began with a very simple conceptual device. They said, well, this is the way mathematical models tend to work. You've got to get rid of extraneous detail and focus on the very fundamentals in order to create a mathematically tractable model. So they said, well, suppose we forget about individuals and all this, and we just think of a pool of genes, which they called the gene pool. And what we ask is, if we know that individuals like humans we're diploid. We got 1 copy of each gene from each of our 2 parents, okay? So let's imagine that we just draw 2 genes from this hypothetical gene pool to form a new individual for the next generation. Now, if we draw those genes at random, what happens? What do we see if we repeat this exercise a number of times? Let's say we draw 100 pairs and put them together. What will be the frequency of the new genotypes? Now, you know the word genotype is the pair of genes you receive from your 2 parents. So to make it concrete, we're going to talk in a moment about the M-N blood group system. You may be MM if you got the M allele from both parents. You may be heterozygous if you got M from one parent and the N from the other, or you may be the little n homozygote. So they imagined that you just draw genes randomly from this gene pool and set up a simple system of notation. Suppose that there's some number, let's call it capital N genes in this gene pool, okay? And there are just 2 types. We're only going to allow 2 different kinds of alleles at this locus, capital A1 or capital A2. Suppose little n one of them are capital A1, all the rest have got to be capital A2 because we're only allowing 2 types. We'll call that little n2, so the sum of little n1 plus little n2 is capital N. It's the total number, right? So we can calculate the relative frequency of the capital A1 gene as little n1 over capital N. That's its fraction, its relative frequency in the population. And we can treat that like a probability. We can think of that fraction, we'll call it little p, as the probability of drawing a capital A1 gene out of the gene pool. This is all written down here. So we think, okay, if we draw 1 capital A1 gene, the probability of drawing it is little p equal to little n1 over capital N. And we'll say that the probability of drawing capital A2 is little n2 over capital N, or we'll call that little q for simplicity. what's the chance that we draw two capital A1 genes in a row to make an A1A1 homozygote? Well, we're assuming we're doing this randomly. That means we invoke the law of statistical independence. So the probabilities are the product of the two probabilities, or P times P, or P squared. Okay? That's all there is to it. Probability that we draw the other homozygote, the capital A2, is Q times Q, or Q squared. What's the probability that we draw the heterozygote? Well, we've got two ways we can do that. We can first draw capital A1 and then capital A2. That's P times Q, using independence. Or we can do it the other way around. We can draw capital A2 first and then capital A1. Again, PQ. So there are two ways, 2PQ for that term. Those are the frequencies of the three genotypes after random mating. Okay, that's all there is to it. And there's a little simple diagram in the book that demonstrates this. So here's your gene pool. It's colored balls. Capital A1 is red. Capital A2 is yellow. What's the chance that we draw two capital A1s? Well, to be concrete, let's suppose that the frequency of capital A1 is 0.7. It's 70% of the total. Then it's 0.7 squared. 0.49 is the probability that we have two capital A1 genes drawn to form a new individual who is A1A1. 0.3 squared is the probability of drawing two capital 2 genes, capital A2 genes. Okay? Two of the yellow ones. And again, there are two ways we can draw the mixed pair, A1A2 or A2A1. Therefore, it's 0.7 times 0.3 plus 0.3 times 0.7 equal in total to 0.42. Okay? So that's all there is to it. It's really easy. Now, this is just another example demonstrating this calculation, but here's the really crucial point. We've done this. We've made this calculation, and we have a new generation that we have created by random draws of these two genes, or these two colored balls. What's the frequency of A1 in the new generation? This is really the key point. What is the frequency of A1 in the new generation after we do the draw? It hasn't changed. It's still 0.7. Okay? And you just work through the exercise, and you'll see that it has not changed. It is still 0.7. Okay, and this just explains this again in a little greater detail, and the slides are posted so you can go through them, but we can calculate algebraically if the frequency of the genotypes are P squared, 2PQ, and Q squared, the frequency of the new A1 gene is all of the A1A1 homozygous class because plus half of the A1A2 heterozygous class. And if we do that, that's P squared, which is 0.49 in the numerical example, plus half of 0.42, which is 0.21. That sum is 0.7. So the gene frequency has not changed. That's the really important result, because it means that under random mating, and incidentally, this is also true for inbreeding and several other kinds of mating, the act of transmitting genes from one generation to the next through the mating process does not change the gene frequencies. Now, it does in some cases if there's sexual selection, if there's what's called assorted phenotypic selection. Let's say that big people like to marry big people, then it can change the gene frequencies. But the typical thing that we observe when we look carefully is random mating. No change in gene frequencies, a conservation of genetic variants. That's the key result, the conservation of genetic variants, meaning that a particulate system of inheritance is exactly what you have to to make natural selection effective because it conserves variance for selection to act on. Very, very crucial point. Okay? So all this is in your book. I wouldn't feel bad about asking you to do a very simple numerical calculation on a quiz question or something, so you should work through it, but I'm sure you've all encountered it before. Okay, well, so this result is due to a man named G.H. Hardy, who was actually a very famous British mathematician. He thought it was a really trivial result and should be obvious to everybody. And the other guy was Weinberg, William Weinberg, who was a German physician who saw this also in 1908 and wrote this paper, which laid the foundation for a field that's now called population genetics. But there's assumptions that they made. Remember I said there's this abstract concept of a gene pool. We've abstracted away the reality that it's individuals who mate, individual genotypes who get together and mate. And we made other assumptions. We assumed that there was no natural selection. We were just tracing what happened to the gene through one round of random mating. We didn't worry about genetic drift. We didn't worry about gene flow or mutation. Just assumed random mating, okay? That's the result is dependent upon those assumptions. Now, what happens if we think about, if we take a step further, we say, okay, this gene pool or abstract may not be good. What happens if we try and make it more complicated and keep track of matings between actual genotypes? Will it still work? Well, I'm here to tell you it will. If you look at it at the genotype level, you know, for this example, we had two genes. There are three genotypes, A1, 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 A2, A2, A2. How many mating pairs are there? You've got to have the genotype of the mother and the genotype of the father. Well, there's A1, A1 mated to A1, A1, and so forth. You can make the list. In fact, if you just think about it, it's 3 times 3, that's 9. But there's some which, you know, if you don't keep track of sex, are complementary. So you can have A1, A1 by A2, A2, or A2, A2 by A1, A1. So if you reduce it, if you don't keep track of the sex of the parent, you can reduce it to 6. Okay? There's 6. Now you say, well, let's suppose we begin with the frequency of any arbitrary frequency for the A1 type. We draw the other mate randomly, and we work our way through the calculation. We actually will find, you will find that the genotype frequencies end up being p squared, 2pq, and q squared. So this abstraction works. Made it much simpler, but if we check it out and do it at a greater level of detail, by keeping track of the genotypes, we still get the same result. So these are the assumptions that are made in doing the calculation. Now let's look at a couple of examples of using this as a null hypothesis. Now, first of all, I want you to think about a slightly more complicated situation. We talked about just 2 alleles, and therefore 3 genotypes. But one nice example is the human ABO blood group series. There are how many alleles here? Well, there are 3, right? A, B, and O. So we're going to have more genotypes to keep track of. How many genotypes? 6. You can write it down. Just take all the combinations. There's AA, B, there's AA, there's BB, there's OO, there's AB, AO, and BO. That's 6. That's it. That enumerates them all. So now we assign frequencies to each of the 3 alleles, and you can ask, do you get an extinction of this Hardy-Weinberg result? And the answer is yes. It works out just the same. If you allow the frequency of the A antigen to be A and the B antigen to be P, and the frequency of the B antigen to be Q, and the frequency of the R antigen to R, the genotype frequencies will be P squared, 2PQ, 2PR, 2QR, R squared, and Q squared. That's it. So this is a good question. Can you extend this? Now, one quick minute. Let's think about looking at the fit of these frequencies to real data. So let's look at real data. This is from your book, so it's in your book. This is the M in blood group frequencies. They're just two alleles taken from different populations. Well, you can calculate the gene frequency of M, and therefore you can calculate the expected genotype frequencies. You know how to do that now, and you can ask, do the genotype frequencies we expect based on random mating look like the ones we observed from the samples? The answer is very close, very, very close. With respect to the blood groups, humans are mating at random. You don't test your prospective mate for their MN blood group. Okay? So random mating really works if you look at it in the real world. Okay, that's it. We'll see you on Monday. Have a great weekend.